Today I have the distinct privilege of introducing Dr. Stout 3 times in a row. So that's great. So Dr. Stout is the Director of Special Pathogens Laboratory and Research Associate Professor at the University of Pittsburgh. A clinical and environmental microbiologist, Dr. Stout is recognized worldwide for more than 30 years of pioneering research in Legionella. The author of more than 200 articles in peer-reviewed journals, as well as textbook chapters on Legionella and Legionnaire's disease. Her expertise includes prevention and control strategies for Legionnaire's disease. An advocate for prevention, Dr. Stout assisted in developing the first Legionella prevention guideline in the United States that has been modeled by national and global health agencies and organizations. She serves on the ASHRAE committee, as well as she's also the board member for the Cooling Technologies Institute. Please join me in giving a warm welcome to Dr. Janet Stout.
Thank you for the kind introduction all 3 times. I really appreciate that.
Give me a second. Start.
There we go. Well, I'd be remiss without thanking Daryl and the organizers for the invitation to be with you at this conference. It's my first time at the Small Drinking Water Conference, and I have to tell you, I've been enjoying it tremendously, and I'm trying to resist the temptation not to drink tap water after listening to all the lectures. But whenever I talk to people about Legionella, I usually have a glass of water and I say, I drink tap water. And the other thing I wanted to thank you for is another opportunity to wear my waterborne pathogen scarf. So on my scarf is E. coli, Salmonella, Crypto, Giardia, and of course, Legionella. So I'm here to talk to you about the role of environmental testing for Legionella and the prevention of building-associated Legionnaire's disease. And my opinions on this topic are informed by many, many years of experience. And just to demonstrate that, I brought out 2 pictures, one quite old from the 1980s, and there I am with Dr. Victor Yu, my mentor, infectious disease physician at the Pittsburgh VA, where I started my work as a graduate student from the Graduate School of Public Health at the University of Pittsburgh. So that wasn't long after the discovery of the causative agent of Legionnaire's disease in 1977. And of course, here we are now, and it gives me great pleasure to be talking to you about Legionella, my favorite microbe. And so when an infectious disease microbiologist studies Legionella for more than 30 years, I thought about this, and I think what that means is that I'm now a Legionellologist. I'm not just a microbiologist anymore. And because of that, what I want to share with you today are just 3 high-level topics. Evaluating the risk of Legionnaire's and Legionella in building water systems, and how and what role does testing for Legionella play in evaluating that risk. And then just touch upon briefly approaches to prevention. And much of what we've learned about prevention has been learned in the healthcare arena, and some of those lessons can be transferred over to non-healthcare facilities. And as you know, with the ASHRAE standard in 2015, what sort of got ushered into the prevention discussion was water management plans, and we'll talk a little bit about that. And then finally, I want to tell you about why Legionella is my favorite microbe. So stay tuned for that. So in terms of how we've been doing in terms of prevention, like what's going to be our report card? Well, unfortunately, we still see many, many outbreaks year after year after year. And those outbreaks are not just occurring in healthcare facilities, although I can say that more and more of the long-term care and nursing home facilities, like this one in Illinois, are sources for outbreaks that had not really been fully appreciated in my earlier years. And of course, commercial hotels. This is an example of an outbreak in Las Vegas. One Ohio outbreak some years ago was related to a brand new facility being erected, celebrating this new building, and then shortly after opening, an outbreak of Legionnaire's disease. And this was related to construction, which is a sort of an underestimated impact to risk of Legionnaire's disease. And in this case, there were 11 cases, 1 death. In almost every outbreak of Legionnaire's disease, there are deaths associated. So that's something that we should all keep in mind. And they did do environmental monitoring, but after the outbreak was detected. So when they did do it, they found more than 3/4 of the outlets positive for Legionella and Legionella pneumophilus serogroup 1, which was the causative agent. And then many of you are aware of the 2015 summer outbreak in the Bronx of New York, one of the largest in history in the United States, 138 cases, 16 deaths. The physicians were successful in isolating Legionella from 26 patients, and the significance of that and frustration for many is that we don't have the isolate to make the epidemiologic link, but in this case they did from 26 different patients. And as a result of that, they were able to use, very sophisticated molecular techniques to demonstrate that the cooling tower on the top of the Opera House Hotel was the source of that outbreak. So in terms of how the incidence of Legionnaire's disease is changing over time, certainly if you'd asked me in 1982, would we still be talking about the increasing incidence of Legionnaire's disease in 2018, I would have said no way. But what reports from CDC and other healthcare organizations show us is that year after year, the incidence of Legionnaire's disease in terms of reported cases goes up and up and up, and it's well over 200% over the last 10 years. So when you look at community-acquired pneumonia caused by Legionella, you sort of start with how many of these pneumonias in the community require hospitalization. And annually, it's about 600,000 in the United States. And studies looking at all pneumonias, what percentage of those pneumonias are caused by Legionella, about 2 to 5%. And so as high as 30,000 cases of Legionnaire's disease are occurring annually in the United States. So as your Legionella prevention teacher, I'm going to have to give us a pretty bad report card today, but have no fear, we're gonna fix that. And in fact, today, the D's stand for something different today. They stand for diagnosis, denial, detection, and disinfection. So I'm going to touch upon each one of these as it relates to Legionella and its prevention in building water systems. So the first point really relates to the diagnosis of Legionnaire's disease. So if a patient sees a doctor with pneumonia, most of the time they don't think about Legionella and they don't do the diagnostic tests necessary, and it will only be done if the physician requests it to get a diagnosis of Legionnaire's disease. So as a result, many cases of Legionnaire's disease are not reported because they're not diagnosed. And this has been shown over and over again, but one of the best studies that I've seen is this report from Rhode Island and Dr. Leonard Mermel's group, where they had a simple hypothesis. If a physician follows the American Thoracic Society and Infectious Disease Society of America guidelines for when to seek a diagnosis of Legionnaire's disease, would any of the cases be missed? And among 37 cases, 41% of the cases of Legionnaire's disease would be missed if the physician sort of followed the guidance from national organizations. So this is just to demonstrate that we see there's underdiagnosis and underreporting. So the problem of Legionnaire's disease associated with buildings is much higher and greater than we thought. Now, in terms of investigations from the CDC, this is a report from 2016, and they reported on 27 different outbreaks that they investigated to answer the question in part, where are these cases occurring? And what they found was hotels. Where are we? We're in a hotel. Anybody have any concern about the water wall that we're walking by every day? Yes, so a hospital outbreak was associated with the water wall. 44% hotels and resorts, 19% long-term care facilities, 15% hospitals, senior living retirement communities. We've seen multiple outbreaks associated with them, workplaces, and in the community. One of the takeaways from this publication had to do specifically with hospitals. Legionella in hospitals accounted for 57% of all the cases, but more disturbingly, 85% of the deaths. So if you get Legionnaire's disease in the community, your chance of death is about 10 to 15%. But if it's within the healthcare environment, it's as high as 30%. So very, very important that we prevent Legionnaire's disease in healthcare facilities, because it really does mean we're saving lives. So in terms of the water systems that we're seeing associated with outbreaks, we have a list here, very familiar to you, and the water systems on the other side of the slide that are implicated as sources for Legionnaire's disease. And I just am underlining hospitals, nursing homes, rehab facilities, because healthcare really is the most important source that we need to be looking for Legionella and detecting Legionella. And so that brings us to the next D, detecting Legionella and building water systems. Now, historically, and I hope this changed dramatically as we go forward, we've had sort of an approach of denial. So here's our ostrich with the head in the sand. And what I mean by that is people are not addressing Legionella risk within their buildings until after cases occur. And in Allegheny County, where in 1993, Dr. Bruce Dixon was instrumental in developing the first guidance document for healthcare facilities, he saw the problem of detecting Legionella this way. And I added the monkeys, so that's not attributable to Dr. Dixon. If you don't look for it, you won't find it. If you don't find it, you don't think you have a problem. And if you don't think you have a problem, you don't do anything about it. And it struck me as so clear and such a wonderful description of the way people approach this problem, I included in every lecture. And what he's talking about, not looking for it, goes back to the early days of what we termed a reactive approach to Legionella in healthcare facilities. So basically, waiting until cases of Legionnaire's disease occur within the healthcare facility, then doing the environmental investigation, testing for Legionella to identify the source. Well, what Dr. Dixon and others in Allegheny County saw as problematic with this approach is that outbreaks after outbreaks after outbreaks were occurring in Allegheny County. And so they developed a proactive approach, which means looking for Legionella before cases occur, determining if the risk is there, addressing the risk to prevent the cases entirely. And so that's sort of been our approach in Allegheny County for years, and is reflected in this 2007 report that we published in Infection Control and Hospital Epidemiology. It's one of the only reports of a prospective pneumonia study looking specifically with Legionella, but also in the environment. So we evaluated over 600 patients in 20 different hospitals across the United States, 14 different states, and over 600 samples for Legionella. And this is what we found. Among these healthcare facilities, if Legionella pneumophila, particularly Legionella pneumophila serogroup 1, was present, and it was extensively colonizing the water distribution system, meaning faucets and showers throughout the hospital were colonized, more than 30% of the outlets that were tested were positive, we saw cases of Legionnaire's disease in those healthcare facilities. And so that's one of the only publications out there that did the parallel clinical and epidemiologic and environmental assessment hand in hand. So what you want to learn from testing for Legionella in a building water systems is where you stand. And there are many, many myths out there about Legionella. One is this one, which I always include. And people think Legionella is everywhere, in every building, and it is not. About 50% of the buildings. Certainly healthcare facilities are at greater risk. So is your facility in the 50% that has Legionella or in the 50% that does not? That is important because it guides clinical decision making. Also knowing what kind of Legionella is in the water guides clinical decision making, because the primary method of diagnosis is the urine antigen test. It detects Legionella pneumophila serogroup 1 only. So what if you have serogroup 6 in your water and you're doing urine antigen tests to find out if you have cases? They're going to be negative and you're never going to know. Now, in terms of the position of CDC, historically it was to wait to test until the case occurs, but their position has shifted over time. Whoops. So this is a quote from Cynthia Whitney, the head of the respiratory disease branch. We are not against testing for Legionella, and particularly in healthcare facilities. So there's changes in philosophy. So the purpose of testing for Legionella in a building water system, particularly healthcare, is to assess the risk. Is it Legionella pneumophilus serogroup 1? How extensive is the colonization? And then that creates the opportunity to control that risk through either engineering controls or water treatment. And you do this before cases occur. Now, whenever somebody does testing, and I appreciated the comment in the earlier session, know what you're going to do before you test, because you have to have a corrective action. Well, many people don't do that, and I often see this panic over a culture result. There's no reason to panic. You're in the same position you were before you tested as after, but it's very helpful to have that roadmap of what to do going forward, and that's the role of the water management plan. So in terms of preventing Legionnaire's disease, again, I want to emphasize that our goal is to prevent the disease. Do what's necessary to reduce Legionella in the water system such that we prevent the illness. That many people try to get to zero Legionella in a water distribution system. It's a complex system, extremely difficult. So I wanted to share with you kind of our experience in Allegheny County, and this is a 2005 report in the American Journal of Infection Control. So we took that guidance document that Dr. Dixon implemented in Allegheny County, which tells people to culture for Legionella, and we looked at the 5 years prior to the guidance document and the 5 years after, and asked the simple question, does this approach reduce hospital-acquired Legionnaire's disease? And so just to really quickly go over what the approach was, you start with culturing for Legionella. If you find it, if there were prior cases, consider disinfection. I don't think this works here, right? If there were no prior cases, then you look at how extensive the distal site positivity is. If it's high, consider disinfection. And if all the answers are no, you just periodically monitor. And what we found in reviewing almost 500 cases of Legionnaire's disease before and after the guidance was that there was a significant reduction in healthcare-acquired cases after the guidance document, and the p-value is admirable. Look at that, 0.0001. So why did the cases get reduced? Well, the reason is they took action when they knew Legionella was present. So 44% in all the acute care facilities in the Pittsburgh and Western Pennsylvania area instituted disinfection technology. And at that time, it was copper-silver ionization. And as you can see, cases of healthcare-acquired Legionnaire's disease were reduced. So again, all the major hospitals, 44%, and testing for Legionella identified the risk, allowing them to prevent the cases and control the risk. It's not just Legionella in healthcare facilities, although it's of greatest concern. So we also looked at Legionella in commercial properties in New York City, 28 non-healthcare buildings. Is Legionella everywhere in those buildings? And the answer is, of course, no. Again, approximately 50% of these buildings were positive for Legionella. You'll be happy to know that we found no Legionella in the incoming cold water. So again, the problem is existing within the building. And the majority of those that were positive for Legionella pneumophilus serogroup 1. So again, just the presence of Legionella serogroup 1 does not mean that there will be cases of Legionnaire's disease, because there are 3 things that have to happen. Legionella has to be in the water, has to be a pathogenic type, has to get into the lung of the individual, transmission, and it has to be a susceptible host. So we know Legionella is present. We want to control it, but doesn't mean it's the same level of risk at every place. We also found that there was a significant association, and this surprised me, of Legionella in the hot water recirculating system of the building and outlet positivity. And this was a significant association. So almost 92% of the buildings that had Legionella in their circulating hot water loop also had it in the distal fixtures. It wasn't 100% predictive. About 27% didn't have Legionella in the hot water recirc, but did have it in the distal fixtures. There was no significant difference, meaning that these parameters were not predictive of the presence or absence of Legionella. No significant difference between positive and negative buildings with regard to free chlorine, outlet temperature, total organic carbon, pH, minerals, and no correlation with HPC and distal site positivity. We talked about that earlier. HPC, heterotrophic plate count, is not predictive of the presence or absence of Legionella. During this study, we also used newer technology, 16S sequencing, to look at the bacterial community. So Legionella is part of an overall community within the system. Sean Ma and Kyle Bibby at the University of Pittsburgh, Civil and Environmental Engineering, were my collaborators. And what this slide shows you, the far left is cold water and then the hot water. You can just see by the different colors of bars, which represent the different phyla, that cold water is very different than hot water. So very different communities. Legionella was found with higher concentration, relative abundance, higher positivity in the hot water, not the cold. So this supports previous research that Legionella is more likely to be in hot water systems than in cold water systems. And then just to reemphasize the difference, we found different community structure in samples that were positive by culture for Legionella, and also different community structure for samples that were negative for Legionella, sort of generating a hypothesis that maybe there's a different community in these areas and that micro niche that was referred to earlier. And this manuscript has been submitted for publication. So in the last few minutes, I just want to reiterate a couple things. This is a quote from Jacob Kuhl's article in 1999, showing that it wasn't the concentration of Legionella in a given site that was predictive of illness, but the distribution. The more likely Legionella is throughout the distribution system, the more likely disease is to occur. How do you evaluate that? We don't want to overcomplicate this for people. So in the Allegheny County guideline, in the New York State guideline, you can look at 10 locations within a building water systems, faucets and showers, and culture that for Legionella. Ask the question, how extensive is the colonization, and is Legionella pneumophilus serogroup 1 present? And as I said, it's now part of the New York State law, which is the first state in the United States to have a regulation. So the proportion of outlets tells you the probability of that resident, patient, occupant, the probability of what I call bumping into Legionella. So we know that the risk is greater. So if you're going to sample for Legionella, how do you do that? Well, the method of sampling is different from cooling towers and water distribution systems. From a cooling tower, sample from the basin, far away from the point of injection on the biocide. Hot water distribution, primary reservoir. Unlike some of the sampling that you do where you flush the sample, you don't want to do that when you're sampling an outlet for Legionella. And the reason you don't is you're going to significantly reduce the likelihood of recovering Legionella, and we actually showed it went down by more than 30% if you do that. This graphic shows you why first draw hot water is the sample that's going to be most likely positive when evaluating a building water system. So Legionella is in that fixture at the point of exposure. There's your biofilm. The water comes out. That's your first draw water. The little bars are bacteria. You flush to waste. Then you take your sample. It's going to be negative. And this has been shown by our group and others to be statistically significant. So in terms of the methods for detecting Legionella, we talked about this earlier in our discussion group. According to CDC, the method should detect multiple species of Legionella, not just Legionella pneumophila, and that means culture. And the International Organization of Standards method 11731 is the one most referenced. And this is particularly true in the case investigation. You need the organism from the environment, you need the organism from the patient. The reason they say detect multiple species is because outbreaks have occurred caused by a species other than Legionella pneumophila. So we have to be careful about that. This is an outbreak of Legionella mcdadii infection. So just to summarize, culture is more reliable than any of the other methods at the current time, including qPCR. And I just want to share with you, this is a title of a poster presentation in the American Society of Microbiology, Julianne Barron, one of our PhD students, basically showing that the 16S ribosomal RNA sequences gave false positive results with other organisms in water, including Aeromonas, Pseudomonas, and Mycobacteria. So that's one of the reasons the PCR for Pneumophila is good, concurrent with culture, but not the genus specific. So as we talked about, not all Legionella are dangerous. What do you want to know? You want to know, is it Legionella pneumophila? There's over 60 species. Not about half of them have caused illness. Highest risk was serogroup 1. And many are common in water, but very rare causes of infection. And then just to wrap up, if you have Legionella in your building, what do you do about it? There are options for disinfection in building water systems. Note that these options are different than disinfection techniques for municipal water. And so sometimes that causes the two regulations, regulating building water systems disinfection, to be problematic. We've evaluated in the field disinfection technology. This is published in 2014 in Infection Control and Hospital Epidemiology. Monochloramine is the newest addition to the toolbox. And what this slide shows you is that above 30% positivity at the beginning of the study, cases were occurring, installed monochloramine on the hot water, reduced positivity below 30%, no cases for now 5 years. So in terms of risk management, we're all sort of familiar with two documents, Center for Medicare and Medicaid Services CMS memo that came out last year and recently was updated. And then of course, ASHRAE guideline, which came out in 2015. This standard is a voluntary standard, not required. It establishes a minimum Legionella risk management approach. There are elements that you should be familiar with: having a team, producing flow diagrams, having control measures, corrective actions, and this one is the most important, demonstrating control. So the only way to do that is testing for Legionella. There are no other parameters that will suffice. So I'm going to finish by telling you why Legionella is my favorite microbe. Now, I don't know if it's because I've been studying it for 30 years, but I've now got a bumper sticker on my car, proud of my highly successful microbe. Legionella, its numbers are up, it's in the news, famous people are talking about it, and it's getting a little paranoid because government agencies are after it. So we saw the numbers of Legionella going up year after year after year. Legionella is obviously in the news that we talked about, Flint, Michigan. There are many organizations that have Legionella in their crosshairs, NSF, ASHRAE, industry organizations. Now, when you start to think about who could possibly be talking about Legionella that's famous, well, Erin Brockovich. She's standing by a chart of cases of Legionnaire's disease. So numbers up in the news, famous people talking about you, government agencies after you. I think Legionella deserves its own star on the Hollywood Walk of Fame. I could be a little biased, I'm not sure. So to summarize, Legionella and Legionnaire's disease is increasing. Potable water distribution systems, not cooling towers, is the primary source, mostly hot water distribution systems. Be proactive in prevention. Include environmental testing for Legionella in your prevention approach. It's the only way to measure. So when I talk to people, I realize that it raises their anxiety. And so I don't like to leave people with all this Legionella-related anxiety. So I have brought medication for Legionella-related anxiety. They are Dr. Stout's Legionella chill pills for Legionella-related anxiety. And I have brought some for you in case you also have raised anxiety. So our goal is to end Legionnaire's disease, and hopefully you will join me in this mission. It's a waterborne pathogen. That's the only route of transmission. So now you get all A's. Thank you. There you go.
Thank you, Dr. Stout. We may have time for just one question. Yes.
Thank you for the presentation. I wanted to see if you had any information that you could share on the burden of chronic fever, because a lot of the stage burden is estimated on Legionnaires of CDC. A few outbreaks have been mentioned chronic fever, but the majority are.
So we've already established I'm not old, I'm experienced. And in my many years of investigating Legionnaire's disease, I personally never seen a case of Pontiac fever. The early, obviously, the outbreak in Pontiac, Michigan, linked the disease in that office building to Legionella because they put guinea pigs in the air ducts, and then they sacrificed the guinea pigs and found Legionella in their spleens. But subsequent investigations, there was exposure to complex water systems with many, many contaminants other than Legionella, and the diagnosis or attributable to Pontiac fever was often made by serologic methods, which are notoriously bad. And so certainly there's an opportunity for more mild infection caused by Legionella, but I don't think there's a lot of Pontiac fever out there.
Thank you. So that brings us to our next speaker, who will be Dr. Helen Buse of EPA. She is in the National Homeland Security Research Center in Cincinnati, and she's working there since 2007. Her research is focused on understanding the role of free-living amoebae, biofilms, water film parameters, and building engineering aspects in the growth of Legionella and Haemophila within drinking water systems. She's currently investigating the disinfection of Legionella and Haemophila within premise plumbing biofilms using various treatment technologies. She has a PhD in microbiology and immunology from the University of Michigan and a BS in biological sciences from Carnegie Mellon University. Please join me in welcoming Dr. Helen Buse.
Thank you, Cynthia, and the organizers of this workshop for inviting me to talk about my research on disinfection of biofilm Legionella. That's been hopefully the result of this presentation will be submitted for journal review in the near future. Just give a little background on our organism. There are currently about 66 species of Legionella, 19 of which have been associated with human disease. Legionella Haemophila makes up the majority of the clinical isolates and is further classified into 15 serogroups based on their reactivity to the Dresden panel of 17 monoclonal antibodies. Obviously, serogroup 1 is the predominant disease-causing serogroup. But like Janet mentioned, there might be some bias to that based on just the routine clinical diagnostic tests that are only capable of detecting serogroup 1, so the other ones may be underreported. Here shows that the incidence rate of the disease quadrupling for the past decade and a half, and with the excluded potable water being responsible for the majority of our rate cases. And in that three-year period, the cases had an associated cost of $9.5 million. So it is a public health and economic burden. And here's an infographic showing the environmental distribution of Legionella in treated water. You see the range next to the green box is derived from culture methods, and next to the blue box is molecular methods, specifically qPCR. And again, like the previous slide, the species associated with human disease is highlighted. And this is the information for distributed water. And we can immediately notice next to the green box is the larger range of Legionella detected in this water type versus treated water. And the higher levels detected via qPCR methods, the values next to the blue boxes there. And the reason why there's two values is just two different standards were used for qPCR. Now, this data was not originally included in my review paper of 2012, but soil is a source of Legionella. And the bottom, the Heidelberg papers are pretty interesting because they identify a very clinically relevant sequence type of Legionella Haemophila in the soil and document their subsequent transfer into rainwater puddles, with the latter being a possible source of Legionellosis. And additionally, with the recent published finding that humidity and rainfall can play a role in Legionnaires' disease outbreaks, it is important just to keep in mind on the possible environmental sources and routes of exposure for this pathogen. Especially in light of this 2016 New England Journal of Medicine correspondence detailing probable person-to-person transmission of Legionnaires' disease. Back in 2014, there was a Legionnaires' disease cluster in the cooling tower complex, heavily contaminated with serogroup 1. Patient 1 was an employee who returned home about 186 miles away, which he shared with his mother. Patient 2, she eventually contracted the disease, and respiratory cultures from both indicated it was the same serogroup 1 strain, same unique sequence type, and identical whole genome sequences. And she was the only one not geographically linked to the cluster. And unless she traveled to that region without disclosing it during the EPI investigation, I mean, this is pretty interesting implications for what we know about disease transmission. But what we are sure about is Legionella pneumophila's stable niche within premise plumbing systems. And you can see the characteristics listed here are commonly attributed to these premise plumbing systems and are usually attributed to also the regrowth potential of Legionella and other opportunistic pathogens like Pseudomonas and mycobacteria. They include high surface areas, volume, amenable growth temperatures, biofilms and sediments, eukaryotic hosts, water stagnation, low to absent residual cross connections, dead legs, nutrients, and the generation of the residual drinking water-derived aerosols. What I'll focus on is the biofilms. So here's just an infographic depicting the stages of biofilm formation, which are attachment, the proliferation, formation of colonies, and then the eventual detachment and downstream colonization of distal sites. I wanted to focus on the initial attachment, which occurs in 2 stages. That reversible attachment, which is dependent on physical forces such as electrostatic charge, hydrophobicity, and van der Waals interactions, and the subsequent irreversible attachment that involves cellular and molecular interactions. Oh. So going back to that first stage of reversible attachment, my EPA colleagues and I, knowing that surface charge plays a very important role in bacterial mobility through the environment and their attachment to these surfaces, we looked at 32 different strains of Legionella pneumophila representing serogroups 1 to 14, and we found that surface charge varied widely between the serogroups, and they were presented as 5 statistically distinct clusters. And this is a general infographic depicting the observation regarding the surface charge of bacteria, their hydrophobicity in regards to their attachment ability. And this study, these studies detailed the bacterial attachment increasing as both a negative charge and hydrophobicity increased, but that strong electrostatic attraction impeded the bacterial growth on these surfaces. So there's a very important balance between their attachment and then their ability to grow and colonize the surfaces. So the same colleagues that I'm helping with this study, we're also trying to further elucidate these mechanisms that allow Legionella to grow in these premise plumbing systems, and ultimately we would like to address whether or not we can exploit these bacterial surface properties, modifying engineering aspects and drinking water quality to negatively impact their survival in these systems. So in other words, can we disrupt their niche? And pretty well documented, the Legionella pneumophila can colonize drinking water biofilms very efficiently. So this project goal was to determine the disinfection efficacy of chlorine and monochloramine, 2 commonly used disinfectants, on these biofilm-associated Legionella pneumophila on common premise plumbing surfaces like copper and PVC. And we used biofilm annular reactors to develop mature 1.5 to 2-year-old drinking water biofilms on those surfaces. The conditions were set to mimic private residential water flow. And we monitored for our various water quality parameters you see on the bottom lines there weekly. And here's just another image of these annular reactors. And you can see in this inner drum slots that were housing these copper and PVC slides to accumulate the biofilm. And I showed here copper slides. The one on the left is new, and the one on the right has a 1.5-year-old biofilms accumulated on it. You can see there's a definite color change and a visible corrosion along the edges. And here's, I don't know how well you can see this, the new PVC, and then you can see kind of like the brownish, light brownish color accumulation on the edges of the slide. Now, after these biofilms were grown, slides were removed and they were suspended in beakers containing about 10 to the 6 CFU per mil of serogroup 1 in sterilized dechlorinated tap water, and they were incubated in batch mode, which was closed for 5 days. After the 5-day period, they were rinsed with sterilized water and then incubated for an additional 24 hours in this water before disinfection experiments. There's just some confocal microscopy showing you the copper biofilm structures on both control, uninoculated, and inoculated slides. What you see in green is just staining of ball cells. It's a stain that recognizes nucleic acid. The red staining shows membrane permeabilized, presumably injured cells. And the blue is an anti-Legionella pneumophila LPS antibody. And the fourth column is the overlay. Now the top row are the control uninoculated. So you can see there's biofilm structures there. Unfortunately, that blue background, you see that copper surface was highly autofluorescent on that particular channel. But on the middle row here at the bottom, which is a zoomed in portion of this white box in the middle row, you can see the biofilm-associated Legionella clustering within these structures. Oh, okay. It's not. Okay. And these are the PVC biofilms, and you can, in contrast to the large diffuse structures on the copper surfaces, the biofilms on the PVC are a little smaller and not more concentrated. And also on the copper surfaces, the biofilm-associated Legionella were clustering within the structures, but on the PVC surfaces, there was really sparse punctate staining throughout the slides. You can see with these blue dots here. And both of the biofilm structures on both surface types were about 30 microns at its maximum. So to compare with the biofilm data, planktonic Legionella, which are the free-floating cells, were exposed to free chlorine and monochloramine at those concentrations under room temperature and pH conditions. And you can see in the black bar, which were the cells exposed to free chlorine, but those very rapid. This is 10 seconds, 20, 30 seconds, and a minute, but very rapid inactivation, which is in accordance with previous studies, whereas with the monochloramine, the inactivation is in the order of minutes. And what is superimposed on top is the 16S ribosomal RNA gene was monitored, the transcript level, so the total RNA was converted to complementary DNA, and a Legionella pneumophila specific assay, which was my heart was beating when Janet was talking about this, and that it didn't work well, but she said it was a genus level, not the species level. I used a species level primer set. With the free chlorine treatment, there was a 1 log decrease pretty quickly from 0 to 1 minute, and then a sharp decrease from 1 to 3. With monochloramine, there was that same 1 log decrease seen but up to 8 minutes. And between 8 and 2.5 hours, there was no significant changes in the transcript levels, which is also consistent with previous reports showing the slow effect of monochloramine on bacterial RNA. And here are just the CT values for 2, 3, and 4 log reduction, again showing a rapid inactivation with free chlorine versus the monochloramine. Here's the biofilm, copper and PVC using free chlorine. You can see the black bars are the culturable Legionella, the CFP persperus enemies, and the heterotrophs, the gray bars. And those are, they were treated for 5, 15, and 30 minutes. So right off the bat, you can see that these biofilm-associated Legionella are more resistant to disinfection than the planktonic free-floating state. After 5 minutes, there's a 1.8 log decrease versus similarly 2 on the PVC surfaces. After 15 minutes, this bar should extend to the 0 because this is total reduction, 2.6 on the copper surfaces versus a 3.9 on the PVC, and 3.1 by 30 minutes, 3.8 by 30. And these are the CT inactivation values at 2, 3, 4 log, showing that Legionella were much more susceptible to inactivation on the PVC surfaces compared to the copper. And that's also you can also see that with the heterotrophic plate counts in the gray bars. There's that initial decrease from 0 to 5 minutes, but the levels were pretty steady, but they were overall lower on the PVC surfaces. And here's the data for the monochloramine treatment, similarly on the copper and PVC. Now, the difference is on the copper surfaces, by 5 minutes, there was only less than a half a log decrease, whereas there was about 2.7 on the PVC surfaces. After 15 minutes, similarly, it was a total 2.6 log decrease, and after 30 minutes, it was also the same. Again, these are the CT inactivation values, showing that with monochloramine, in contrast to the free chlorine, the Legionella seemed to be more susceptible on these copper surfaces versus the PVC. And there was no difference between them, the heterotrophs in these biofilms. And these are the same exact bar graphs shown on the previous two slides, but with that, the qPCR data superimposed on top. The triangles are the gene levels, the total gene levels assessed by total genomic DNA extraction and then followed by qPCR. The circles are the transcript levels assessed by converting the total RNA to cDNA. You can see that the gene levels from total DNA were generally higher than the transcript levels. And free chlorine treatment of the biofilm Legionella impacted the gene and transcript levels from 0 to 30 minutes, up at the panel A and B, up at the top. And for copper surfaces, that's panels A and C, overall, the transcript levels were lower compared to the PVC biofilms. So that means that the copper surface alone has a negative impact on the transcript levels of Legionella pneumophila. Now, before I go on to my summary points, I wanted to discuss some other studies that are relevant to this project. This one actually, group grew 3-year-old drinking water biofilms on stainless steel, and they showed a maximum thickness of 30 microns on 1.5-year-old biofilms. And over the course of that 1.5 years to 3 years, the average thickness of these biofilms were 14 microns. And they actually operated their reactor under less turbulent flow conditions, but there was no stagnation, whereas in this study, because we were modeling private residences, there were stagnation periods during the day and at night. And this study developed Pseudomonas aeruginosa and Klebsiella pneumoniae biofilms on stainless steel. They used rich growth medium and only grew them for 1 week in batch mode, generating 150 to 200 micron thick biofilms. But after 30 minutes of 2.5 ppm free chlorine exposure, they found that 70% of the free chlorine was able to penetrate the biofilms to a depth of 50 microns. Their disinfection conditions were not stated. And this study was performed by my colleagues in ORD. They grew nitrifying biofilms that were 6 months old on polycarbonate, generating 200 micron thick or greater biofilms. And they found that with comparing free chlorine and monochloramine after 30 minutes of treatment, about 8% of the bulk water free chlorine and about 15% of the monochloramine was able to penetrate the biofilms to a depth of 50 microns. Again, the pH was 8 and 23, similar to the conditions I used in my study. There was very low disinfectant demand on my biofilms during this experiment, and I didn't measure the penetration of these disinfectants, but these previous studies seem to show that there is some penetration, especially up to the 30 micron thickness that were measured in the biofilms from this study. So on to the summary points. Obviously, the planktonic Legionella pneumophila using free chlorine was more effective at inactivation compared to the monochloramine treatment. And for biofilm-associated Legionella, monochloramine was more effective on the copper biofilms, while free chlorine was more effective on the PVC biofilms. So that was pretty interesting. This is just an infographic showing the CT values at the 4 log in inactivation. And also, free chlorine treatment negatively impacted the gene transcript levels and may act synergistically with copper surfaces to further reduce their levels. And I'd like to acknowledge the people involved with these projects, Dr. Jeff Szabo for his engineering expertise, Dr. Dave Wahman for his chemistry expertise, Brian Morris and Ian Struey for his technical assistance, and Chet Clausen at the University of Cincinnati Microscopy Corps, and then also my ORD colleagues Darren Lytle, Jill Hawley, and Christy Mullen, and also Drs. Brian Raphael and Marty Solon for providing some Legionella pneumophila strains for testing. And with that, I would like to take any questions you may have.
Thank you, Helen. We may have time for 2 or 3 questions. Are there any questions on the webinar? I forgot to mention that this session is being broadcast on a webinar, and I think there are questions from the webinar.
This is
from the webinar. So was ammonia free and total done on the analysis to determine the potential contribution from growth bacteria and was nitrate?
Is this during the development of the biofilms, or was this during the disinfection experiments? During the disinfection experiments, total and free chlorine as well as monochlorine was monitored throughout that exposure period. And for the biofilm development, yes, we actually measured chlorine decay between the stacking periods. So the engineer, Dr. Jeff Siegel, and I, we kind of said, well, to mimic a private residence, we had the water flowing for 2 hours in the morning, and then when everyone's at work or the kids are in school, there's there's just stagnation, no water flow, and then at night there's 4 hours, the water flows turn back on for 4 hours, and on the weekends it's similar, but with a 2-hour run in the middle of the day. But we did monitor for chlorine, so these are actually drinking water biofilms that would be growing in your normal household plumbing.
What was the pH in the experiments?
The pH for the disinfections were at 8. The water that we're feeding the reactors were around 8 and 8.5. Normal tap water.
Do we have any other questions?
I have one.
Yes.
Sort of historically, we compare kind of bioinformation of Legionella with another organism, Pseudomonas aeruginosa. Legionella is kind of wimpy by comparison, doesn't create like a luxurious biofilm. So in your biofilm model, you mentioned that you measured maximum thickness and that was average thickness. Was it uneven? Was there spotting?
Yes, definitely. The 30 microns was the maximum thickness. You can see those really bright green structures, but yet there were areas of the surface of the slides where there were basically no biofilms. And you can see it more clearly with that PVC image I showed. There was that lightish brown material on the edges of the PVC, but for the most part, the middle of the PVC slide was still pretty white, like the new one on the left-hand side. So there's definitely just some heterogeneity in the structure.
I had a question for Dr. Stout. Your research showed that there was no correlation between Legionella presence and some of the elements like manganese and things. What are your thoughts on iron? Because now I think they believe iron is involved in as nutrient for Legionella.
So going back to early studies in the 1990s where we were looking at Legionella in home water systems, we sampled over 200 in the Pittsburgh area where we found obviously large buildings like hospitals, a lot of Legionella, but only about 6% of the homes on average had Legionella in the Pittsburgh area. But in the one community where we found a higher proportion of homes positive, we found a higher concentration of iron. So we speculated because of the nutrient requirement of iron pyrophosphate that the presence of iron was actually helping Legionella in the environment. And of course, that was sort of suggested in the grant as well.
Dr. Pius, University of Massachusetts, both either or both of you comment on source of Legionella. Originally, you're documenting water systems and processing as an environmental engineer, from a source control perspective. I understand prevention, but what's your thoughts on the source? You said not ubiquitous presence, but is it ubiquitous source of the environment?
Take a shot at first. So going back to the 1980s, Stan States and others at the Pittsburgh Water and Sewer Authority looked at the source water and were able, through concentration of large volumes of water, to show very, very small amounts of Legionella in the source water. And in fact, at the Pittsburgh VA, when I was a master's student, we took water from the large storage tank. We put it through heat coils and things like that. And it was very difficult to grow any, but we did. So we and others have said that Legionella is coming into our buildings in the cold water source more often than not detectable in that water. In terms of the sources, nationwide, and as it was pointed out earlier, groundwater as well as surface waters have been implicated, shown to have presence of Legionella after these ways of detecting Legionella, but not all of them. And certainly this group, more than any, appreciates the diversity of water sources and water quality. And so Legionella is not in all of those source waters, but it's in many of them, and that's where Legionella is coming from as it enters our buildings and devices.
Here's another webinar question. Before I ask, apparently the webinar people can't hear this mic or the table mics. So they can only hear the podium mic. So this is a question for Janet and Helen. With regard to mitigation, there are two questions that you throw together. So what's the effectiveness? Has anybody said the effectiveness of point of use treatment devices? And has anybody looked at flushing periodically, say at night or on the weekend, flushing a hot water line with cold water that has a residual presence? And how does that work in controlling it?
So just to repeat the questions, just comment on point-of-use devices as a mitigation strategy. And then the second one is, what is the effect, or has merely flushing outlets been shown to be effective as a mitigation strategy? So point-of-use filtration is and has been used. These are 0.2 or less micron filters that are, there's an adapter on the end of the faucet and shower. They click on. They are a physical barrier for Legionella. We've published 2 separate evaluations of point-of-use filtration, and in each of them, the period of time specified by the manufacturer, the first one in the early stages was only 7 days. Now it's up to more than 60 days of use. Completely rid the water coming out of the faucet filter of Legionella. Now, some other very small organisms did make it through over time. So the idea is that it's not a sterilizing filter. There are some things that will come through, but Pseudomonas, Mycobacteria, and Legionella were excluded. Believe it or not, in our first early days of trying to battle Legionella, we tried just flushing water through the fixtures. And so I can tell you from personal experience that will not work in and of itself. And part of the reason is even if you had a detectable residual of chlorine in the cold water, it's not sufficient for disinfection of Legionella. Legionella requires higher concentrations for control, and there's basically no disinfectant in the hot water. So if you're just moving the water, remember Legionella is attached in the fixtures up and through the nooks and crannies, and it's just going to come back. You might have a temporary reduction, but it's really going to be there and coming back.
So even if you held the cold water in that lateral or in the faucet fixture, like over the weekend or overnight, still that doesn't work.
That's correct. There's not enough disinfectant in that water to control Legionella or kill it, especially in these fixtures where there's accumulated biofilms of various natures, lots of organic material, lots of metals that would interact with chlorine and disinfectants. So it's really the sort of the tried and true. It has to have a high enough concentration long enough.
All right. Thank you very much. That brings us to our last speaker of this session, Dr. Jay Garland. He's a division director in ORD of the Office of Research and Development in EPA, and he has a PhD in environmental science from the University of Virginia and over 20 years experience working on NASA's efforts to develop closed biogenerative life support systems for extended human space flights. So he could be our next person for the Space Force, I suppose. NASA recognized him for creative technology innovation on 4 separate occasions. He has authored over 100 scientific papers on a range of topics, including methods for microbial community analysis, factors affecting survival of human-associated pathogens and various biological approaches for recycling waste. His work is committed to the development of sustainable systems using sound ecological principles and innovative technologies. Please join me in welcoming Dr. Jay Garland.
Okay. Thanks, India. Thank you, everyone. Just a full disclosure. I don't agree with the Space Corps. No. Second disclosure is I'm not going to mention anything about Legionella or even really about drinking water strictly. So if you want to leave, you still have time. But I'm going to talk about on-site reuse, mostly for non-potable applications. So hopefully this title of emergence, what I mean by emergence, you'll understand. First off, I just want to recognize a number of people that helped make this work. I mean, the top bullet there is all the people that are in EPA ORD with me working on this project. The second bullet, Paula Kehoe, and the entire, what we call the Blue Ribbon Commission on On-Site Non-Potable Reuse. This is a group of utilities and public health agencies from states and utilities across the country interested in making, implementing this technology. So they've been very important in defining the research, and I can't thank them enough. And then a number of different collaborators, both in universities and in private business. So my extraterrestrial background, okay? So this is what I used to work on. I used to work on these life support systems for lunar base or Mars base, and you can see that they were all just artist renditions, right? Because we haven't done this yet. I left NASA about 7 years ago, and this is what I used to show then. Now, since I left, I guess there was this new movie, right? The Martian with Matt Damon. So this is a much sexier version of what I used to do. And to tell you the truth, what Matt is doing there in that movie, he's growing potatoes on the fecal matter of his ex crewmates. And I've done that. That's the one thing I have in common with Matt Damon. But the point, why am I talking about NASA, right? Other than bringing up what I have in common with Matt Damon, maybe that's part of it. But the other part is this quote kind of represents this. If we knew how to live on Mars or on the moon, and you don't have ecological services to rely upon, you have to design from the beginning regenerative systems. You have to think about regeneration from the get-go. And that, to me, is the most important design concept I learned when I was with NASA. And that's why I didn't change my view when I came back to Earth. I just came back to Earth because I thought it was more important to do the work here than in space. So as an example, I mean, and I know this kind of sounds like, oh, your waste is your resource. It's some magical, kind of a process. It's not violating the laws of thermodynamics. It's about design and about really rethinking how we approach what we produce and what we consume. As an example, you can think of a building as a production source for water. It produces water, right? You could view it as, all right, I got all these water. I got rainfall on the roof. I've got precipitation at grade. I've got nuisance groundwater collecting around the building. I've got wastewater from clothes and laundry washing and the toilet water. I've got all these wastes I have to deal with. Right? And you do have to deal with them, but if you change it around, it's not the problem. That is the solution to some of your issues with supplying drinking water sustainably. If you could recycle these waters for maybe not potable use, but non-potable use, you can greatly decrease the demand for water. So that's why I bring up NASA, not because I think it's more important. I am convinced that what I do now is much more important. So, emergence of these systems. One caveat is that when I'm going to talk about how this is increasing, but it's not that new. It's been around. The Grand Canyon Village in the 20s, in 1926, was collecting, treating their wastewater and reusing it to flush toilets. So, but more recently, I just want to show you some examples of this. This is the Solaire building in Battery Park, New York City. They have a membrane bioreactor in the basement of the residents, and they treat all the black water, all the wastewater from that building, treat it and reuse it for toilet flushing, cooling, and irrigation. And that's been operating for over a decade now. And the real driver in this case, you say New York City, what they don't, not short of water, but what they have is they have a high sewer tax. So this decreases the amount of money they have to pay for sewers because they're decreasing the flow because they're recycling some of that waste. San Francisco Public Utilities headquarters, they're leading a lot of this effort. they have a rainwater harvesting system. They also do wetland treatment and further treatment of the wastewater for toilet flushing at about a 5,000 gallon capacity. And so that is their system outside, part of their system, kind of a green space. There's a couple, San Francisco has implemented this as a requirement for new buildings to do non-potable reuse. So there's a number of different projects emerging there. One is the 181 Fremont building, which is coming online this year, and they have a 5,000 gallon gray water treatment system. And it's a mixed-use building, both residential and office building. And their big driver, a lot, was sustainability goals and LEED goals for the building. So not necessarily money saved, but more about being able to demonstrate their environmental leadership. Salesforce Tower, which is also in San Francisco, is the second largest building west of the Mississippi, is coming online this year with a 30,000 gallon per day membrane bioreactor treatment for the black water, for use of flushing, toilet flushing, irrigation, and cooling. And their drivers were also related to sustainability and LEED. There's in Portland, Oregon, Haslo on the 8th is this that's their treatment system. They light it up at night so it's colorful. That's why it looks pink. Their wastes are not pink. But they have that treatment, and they recycle a lot of it through vegetated areas in the property. So it's part of their driver was to actually produce this aesthetically pleasing landscape with the water. All right, so there's lots of examples of this emerging. 4 years ago now, San Francisco, SFPUC, hosted a group of people interested in trying to advance these systems. So as this map indicates, it was from around the country. It's not just a Western issue. There's not just a Western interest in this. People from across the country. And this group has expanded. And like I said, it's either utilities or public health agencies. People are going to have to permit these systems, or people are going to have to deal with them. So they came together to say, what were the issues preventing further adoption? Now, the other thing they talked about is why are they interested? And and I touched on some of these, but it's a variety. One is that it's water scarcity. Clearly, if you can recycle the water for non-potable uses, you're putting the strain off your drinking water supplies. That's clearly what San Francisco is mainly interested in, because they're worried about the longevity of the Hetch Hetchy water system. But there's also, if you're going to do that, there's also interest in the efficiency. If you can treat the water only as much as you need it, don't treat everything to potable standards, then that might reduce the cost of treatment. And if you also treat it on site, then you're reducing the need for these large redistribution systems, the purple pipe networks, which are very prevalent in, say, Florida. Thirdly, kind of other areas that, if you could decrease the pressure off the drinking water, you can defer capital costs in these large-scale projects. Don't have a large-scale advancement for increased drinking water because you're increasing efficiency. You can also, like I mentioned, in New York City, reduce pollution and loading the sewers and water bodies, and also reduce stormwater if you're collecting that. And then, as I mentioned, there's a lot of interest in, well, this one I hadn't mentioned, this idea of resiliency and adaptability of our water and wastewater, the idea of having more of a distributed or nodal distribution to your water structure. I mean, that's again, something, talking with San Francisco, 30 years down the road, they're thinking about what is their system going to look like? Are they going to have 30 different wastewater treatment systems? They're going to have nodes that are that size as opposed to just one centralized system. So on site, but kind of expanded. So there's this idea that you have multiple systems give you more resiliency. And then finally, as I mentioned, there's interest in the city in generating environmental amenities in the urban corridors, green space, and meeting these kind of LEED standards. So there's a lot of different drivers for why this is happening. But this group, when they met, the key thing was, the key outcomes of that meeting, and what we've really been working on the last 5 years, is, okay, we need local management programs, but we need local management programs that tell us what do we have to monitor for, and what is the sufficient level of treatment, and how do I know I'm there? Right? There really is no standard guidance for this, and that was preventing their adoption of these systems. So we developed a research program in ORD around helping them answer that question, right? And this graphic kind of shows, I know we're not finding any new water, we have all the water on the earth, but it's finding new sources of water, right? So in this case, looking at these various types of water that I mentioned, rainwater, gray water, black water, storm water, looking at the treatment for non-potable use. And the three main questions are, how do you define acceptable treatment? How do you know you treated the water so you can use it safely? Second thing is, how do you monitor that you're continuing to do that? And the third question, which I really don't have time to talk about today too much, is about, does this make sense to do? Now, clearly what I just said, there's a lot of drivers. It makes sense for a lot of people doing this. We've done some life cycle assessment analysis of these systems, these kind of building scale systems versus, what's the cost of that versus the benefits, i.e. the reduced drinking water that you're supplying and the cost of that. So we've done those kind of analyses, and I can share them with you. The bottom line is that if you look at the broad perspective, you look at the cost, the avoided cost of drinking water supplied to you, you can operate it. There is a net benefit, right? So, but let's talk about this question of how do you know how well to treat it. For gray water use in the flush toilets, there are a number of standards out there. A lot of states have standards for things like BOD, TSS, and I'm going to focus most of the conversation on microbiology. So there's total coliform and E. coli levels, but you notice in that box there's variability from state to state. So that's one of the challenges. They're inconsistent. Which one's right? So more recently, NSF, the National Sanitation Foundation, they did develop 350 water quality for gray water use for toilet flushing. So this is great. They have standard levels so that everybody can adopt the same standard. So that's an improvement. The challenge, though, is that it's not risk-based. And what does that mean? Well, what it means is, what does having, what does having 240 NPM per 100 mil E. coli mean in terms of risk for use in the toilet flush? Where does that number come from? And I got to tell you, it really is largely kind of a transferring numbers from one application to another, and it really isn't risk-based. It's not telling you anything about the risk for that particular use. The other thing we were very interested in, the examples I've shown you is just for gray water use for toilet flushing. We wanted to develop an approach that could be used for all those waters that I mentioned, whether it's rainwater, gray water, black water, same approach to assessing risk and the level of treatment for different uses. Could be toilet flush, could be laundry use, could be unrestricted irrigation. So really trying to have a more flexible approach. And that's important because, if you're looking at rainwater, you're not going to be worried about E. coli, for example, necessarily, right? You're not worried about human fecal contamination. You're worried about other kinds of contamination. All right, so let's see. So what is this idea of risk-based? What does that mean? Well, risk-based means that you have a specific level of protection, like 10 to the minus 4th infections per person per year, that you're trying, this is your standard, this is acceptable, and you need to treat the water where you're not increasing risk above that level. So you have a reference point of acceptable risk. And WHO does this for risk-based targets for wastewater reuse for agriculture. State of California, who's doing a lot of potable reuse, they also, this is the same approach, define an acceptable level of risk first, and then develop a process to define that risk. And I go back to NASA here for a second, because largely this idea of having a very structured risk assessment approach around microbial risk comes from NASA back in the 60s, because they wanted to answer the question, how safe is the food for the astronauts? They didn't want them getting sick in flight, so they developed what at that point was called a hazardous analysis and critical control point analysis, where you really systematically looked at where the risks were, what are the risks, where are they, try to quantify it, and then mitigate in response to that so you can minimize the risk. And this approach was really widely adapted in the food industry and more recently in the water industry. So this idea of passive, it's also kind of emerged into quantitative microbial risk assessment, which is really what we've applied to this problem, right? So in this case, you've got same diagram, various sources of water. You got to treat the water for a given use. So on your right-hand side, the things you want to know about is what volume are people getting exposed to, what are the concentrations of the pathogens in that water, what's the dose response for that pathogen it enters, and then you can estimate what your predicted risk is if you use that water, if you don't treat it. But then you have a log reduction target that brings that level down to an acceptable risk. So bottom line of this whole approach is defining LRTs, right? The log reduction targets for different waters for different uses. And I could spend an hour talking about that model. You know, how do you predict the levels of pathogens? And we've developed models to predict what we expect the pathogen concentration to be in gray water and black water of different size buildings, because it depends on how many people are in the building, because it depends on the infection rates. So our models estimate infection rates and fecal load and translate that to pathogen concentrations. And then also we try to harvest whatever available data there is on some of these. And I got to tell you, that is one of the advantages of doing this QMRA, is it structures your thinking, so it defines where you really have gaps in your understanding, where are the gaps in the knowledge. In this case, I can tell you for rainwater, there's a real lack of data on pathogens in rainwater for the US. It's lacking. We're doing some work now in collaboration with Colorado State University to increase that. Another one is stormwater. Stormwater, when we tried to model it, the numbers are all over the place. There's not good numbers on pathogens in stormwater. So that's another critical area for research. But anyway, the end result of this work is that you get this table of, and it's kind of a big room, so it's hard to read, but this is data that we provided. We published this work, and it's gone into this document, and this Blue Ribbon Commission has rolled out this framework document for regulation at the state level for these different non-potable reasons. So this shows you the actual log reduction target for enteric viruses, protozoan bacteria for a given type of water, domestic wastewater, gray water, storm water, and roof runoff. And the numbers that you see there are the log reduction. This is what you have to define a treatment system to meet that log reduction. And the numbers in parentheses versus the numbers not in the parentheses, those are, you notice the ones in parentheses are slightly smaller, and that's for the lower level of acceptable risk. I showed you two acceptable level risks, 10 to the minus fourth and 10 to the minus second. 10 to the minus fourth is what we use for drinking water. 10 to the minus second is what we use for recreational exposures. Non-potable use, you know, it's somewhere in between, right? So we modeled it both ways and left it kind of up again to the local authorities to define what they want to use for their particular guidelines. So that's the QMRA side of things. The second question is monitoring. You know, the idea here is you designed a system to meet the risk-based performance targets, right? That's what this whole beta based on. The LRTs that we define, that's what you use, the valid design system to meet that treatment requirement. But then how do you verify that performance? Well, what this group, and again, it's in the documents that I'll show you at the end, a reference to all these documents from this Blue Ribbon Commission group, emphasizes routine monitoring of some, typically what's done is routine monitoring of indicator organisms, but that really doesn't provide real-time risk-based information, like I've just explained. And I'm going to tell you in a second too that it doesn't necessarily, at least given the current indicators we use, they're not good enough. They're not high enough levels to give you the dynamic range you need. So what this group has really emphasized is that look at operational monitoring. Look at what you can monitor real time, whether it's transmembrane pressure or UV fluence or chlorine or disinfection residual, whatever you can measure online in more real time, use that to validate your system as a surrogate is performing the way you've demonstrated in your original validation studies that it works. And you rely on those predominantly because that's really what's going to protect public health, because that's going to be real online measurements. So the surrogates for LRTs. Now, but you're also going to have to, at startup and commissioning, and also after it goes out of spec, you're going to have to validate it. You can actually go out and say, yes, this is removing X logs of viruses, protozoan, or bacteria for you to be sure of its performance. So how do you do that? How do you do again, the reliance is on, rely on your online monitoring as much as possible, but when you have to measure a biological reduction, what do you use? Well, you could measure pathogens themselves, but it's difficult. There's lots of different pathogens. They're very sporadic in occurrence, right? So you could be measuring it today, and it could be 0, but that's because you don't have infected people today in your small building, right? And your system really isn't performing well, so tomorrow it won't be safe. So you need something else that's there more consistently. So you can measure biological surrogates that represent the pathogens. The typical surrogates, like if you're talking about a typical bacterial surrogate, it's a fecal indicator organism. I'm going to tell you those are too dilute. I'll show you in a second. And then you could spike with some surrogate and then calculate the reduction. And this is done. MS2 as a virus is used often to spike and look at reductions, but that's difficult too, because you got to grow up these organisms and do challenge testing. It's done, but one option that we're looking at now is trying to look at endogenous microbes as alternative biological surrogates. And what I mean by endogenous organisms that are things that are there anyway. They're there in high numbers. So our approach to this endogenous biological surrogates, step one is look at what's there, what microbes are present, what are at very high levels, high enough levels above what your LRT is so it has a dynamic range to use as a monitoring tool. And I put this in there now, but the reason why this is so possible now is that we live in what I call the age of the microbiome, right? We live everywhere. If you look at stories, you hear stories about microbiome all the time, in you, on you, in what you touch, in what you rode today, wherever in your house, right? We realize that the world is full of microorganisms, right? You yourself, each one of you walking around, are more microbes than you are human cells, right? You got 10 times more microbes than your own cells, and those microbes are very diverse, and they have 300 times the number of genes in your cells do, right? So you really are, and some would argue that you're only up walking around because microbes evolved into multicellular animals because they themselves wanted to get up and move around, the microbes. But when I say, that was a joke, okay? I say we live in the age of the microbe because we realize the importance of microorganisms in human health, but the age of the microbiome has been the age of the planet because the microbes have been around since the inception or near after the inception of the planet, and they have really created the biosphere and drove the creation of life on Earth. So, so anyway, age of the microbiome. But you do that, and I'm going to show you some results on what we've looked at in terms of what's there in wastewater. The second one is, what are the, once you've identified what they are, how abundant are they? They must be at or above the LRT, and they must be consistently present in the influent. You want them always there if you're going to use them as a monitoring tool. And so if you find things that are at high level that are consistently there, then you would then target and develop actual methods where you compare that log reduction of that given target to the actual pathogen. So this is the stepwise progress. So what I'm going to talk to you about today is really in that first and second. What's there and at what level are they? Okay. So real quickly, what I'll finish is that if you look at this graph, this is for gray water. You've got on the left-hand side, those 4 bars are human-specific markers, and fecal markers, like E. coli, Bacteroides, human-specific Bacteroides, and enterococci. Those are the levels in gray water. The ones on the right-hand side, this is laundry water that we collected from the University of Cincinnati. They're much higher, and those are all things that are associated not with human feces, but human skin, right? And if you look at what the log reduction target is that we're trying to meet for this use, you would have to have an organism in your influent that's at least above that level. So the point of this graph is that the typical markers, like the fecal indicator markers, are not high enough in the influent to give you the dynamic range to measure them in the effluent. So the other ones you potentially could, these skin flora, but it gets even more complicated because this graph is a deep sequencing analysis of the microbiome of gray water. And on the left-hand side, in the column that says laundry, that's the sequencing of all that laundry water that I talked about from University of Cincinnati, which was directly out of the laundry machine. And what you see there are, and you can't read these names on the side, but they are all human-associated skin bacteria. So it makes sense, right? They dominate the gray water there. Maybe those are good targets. But if you move to the right, all of those different, the nomenclature bottom, they all represent water that we collected from showers from where they're recycling it in Colorado State University. And that water, once it's passed through the drainage pipe or the collection pipes, the human signal, the human skin signal is gone. What you see is a number of other different organisms that are associated with anaerobic microaerophilic sites where there's a lot of active biodegradation going on. So it's the pipe microflora, it's the infrastructure microbiome. Those are the organisms that might be what you'd want to target for looking at log removals for bacteria. It gets even more complicated when you look at viruses, and we started to look at viruses. this just shows you we start doing deep sequencing of viruses in wastewater. 79% of them are not even described, right? So it's but of the 21% are described. You see a number of different names there, and we've started to target some of those specific targets in wastewater, right? Some of this on-site collected wastewater. And again, here's some graphs of these different viral targets in gray water with these lines. The solid red line represents where the level would need to be effective at the 10 to the minus second risk level, and the dotted line is where it would have to be to use as a log reduction target, 10 to the minus fourth level. So some of these new targets look pretty good. Some of them are not sufficient. They're not high enough levels. That's for on-site collected wastewater. Here's one for gray water, slightly different, same set of organisms, but different mix. So to give you the idea, we're looking at these new targets and evaluating what their frequency and abundance is with some very promising candidates. So what the hell did I just say? The first thing is that we're in the early adoption phase of on-site reuse. I mean, there's a variety of factors, a variety of drivers. The cost benefits are favorable now for a lot of places. And the further demonstration need to provide real-world data, right? I mean, that is going on. And the data that I just showed was from collection of on-site collected wastewater. But in order to protect public health during these demonstrations and with wider adoption, emphasize risk modeling to define the treatment and new approaches to monitoring, both which involve modeling and mining the microbiome, trying to understand what's there and quantitatively understand what using these new techniques to look at what surrogates you need to monitor. So anyway, thanks. Follow up the resources for on-site non-potable programs at that uswateralliance.org website will provide all the documents that I've just referred to that this group has produced, as well as the references to the peer-reviewed work that drove some of the numbers that I mentioned. So thanks for listening to something different.
Thank you, Jay. There's time for a couple of questions. Any questions from the audience? Tom, do you have any webinar questions? So I'm given to understand that the speakers cannot be heard on the webinar if you're sitting there. So if you could please come here and answer your question, Jay. Thank you.
So gray water is very variable depending on the source, location, other factors. How does that impact the setting of removal goals? How does that impact the use of these indicators? So the question was related to the variability in gray water. I mean, one issue is the variability in the pathogen levels in gray water, and so to that end, we have modeled different scales of gray water collection systems, both size and type, whether it's laundry or whether it's shower water. And what drives the differences is the amount of fecal loading. So we use that as a surrogate to estimate what the fecal loading is, then multiply that or factor in the level of what the expected infection rate is, and that gives us, it allows us to model the different concentrations of pathogens in the gray water. Now, the second question is, and what I showed is, if you look at the composition of the gray water, both bacterial and virally, when you want to develop surrogates, you're right, it does look to be variable. For the bacteria, what seems to be the primary driver is whether you've collected the gray water. Most gray water collection systems, you store it and equilibrate the volume before you treat it and reuse it. So what you really want to know is what's in that storage tank, and what's in that storage tank looks much like the right-hand side of the graph that I mentioned, these dominated by infrastructure organisms. In terms of the viral load, we do, for those targets that we looked at, there was a lot of variability between the different targets. So we looked at the things that the organism that was highest, consistently highest between those, you want to try to pick something that's commonly high, was, and this is kind of unexpected, it was the pepper mild
model virus. This is a virus that attacks peppers, but it's at very high concentrations in wastewater. And for the data that I showed, it may not have been apparent for those graphs, that was the highest, the virus target that we looked at that was at the highest level for both gray water and black water sources. So the response to the question, do you want to try to find what, given there's variability, but are there common things at high levels that you could use as a surrogate? And to date, it looks like this pepper mild model virus is maybe one.
Maybe I missed it, but you said it maybe. When you are talking about pathogens, you are talking about viruses and bacteria. What about the other kinds of pathogens? You have to have some goal, some definition of that, and that's another kind of protection.
Right. The three classes of pathogens of concern that we modeled were viruses, protozoa, and bacteria. So the one table that I showed and the results we produced have log reduction targets for each one of those broad classes of pathogens. So that is one of the approaches using QMRA, is that you kind of look at what the different groupings of pathogens are, not every single pathogen, but you look at the different classes and then pick what you think is the representative pathogen of most concern within that group. So for when we've done the modeling for gray water and black water, for example, norovirus is the one that we are most concerned with. It's at the highest levels and has a very low infectious dose, so it's of most concern. So that's what we model as the log reduction target of removing that. The assumption is if you've done that, then the other organisms are going to be removed as well. But bacteria behave differently, protozoa behave differently, so you need to also include log reduction targets for those classes, and the data do show that. But as classes, not as individual, different kinds of.
My question was, you showed potential organisms that you use in terms of bacteria and viruses, but I didn't see anything about protozoa.
Yeah, you're right. So we've modeled the LRTs for all three different classes, and of the in terms of the monitoring work, we've done some work with bacteria and looked at the viruses, the virome as well, but we have not to date started looking at the protozoans. So in terms of monitoring, we have to still rely on I guess what the accepted current practices are.
All right. Thank you very much. This brings us to the end of the session. Let's give us a big round of applause to our speakers. Thank you very much, and thank you to the webinar folks for your interest, and we continue to bring you monthly webinars. So thank you. See you at 3:30.
And the moderator. Oh, thank you. It was nice, very good talk. So you might want to give this to your next.
Yes. No, I have plenty. You can keep their mitts. Their mitts, yes.
Cool, I'll take